predictability. So fail-safe. You know, when you look at the different industries, across industries, electronics industry, aviation industry, automobile industry, everybody's working at a very high level of confidence in the product, right? We board the plane, we don't think about what might happen. You know, 20, 30 years ago, people were worried. Now, aviation industry is the safest industry because they operate at a Six Sigma level. 1 in 10 million accidents happen. In pharmacy, 1 in 3, maybe more, has side effects. We can't really deal with this. But I think we will, if we apply ourselves. And unfortunately, I think the drug development business was victim of premature development. In the 1990s, when software and hardware were primitive, people started using this for drug design and failed because of the fact that this was primitive. And now, 2 decades later, I think we are at the cusp of, in my opinion, a major revolution. And people who triggered this, like Chris Lipinski, they admitted in review papers that, in fact, they created a monster in a way, and big pharma CEOs and executives doubted the power of computers. They said it's a very expensive way of making hot air. Now I think this will change. So I will say a few words where I think the world is going in this, and I'm trying on a very small scale to move this frontier a little bit forward. And with IBM's help, we are moving in this direction, but it's a major undertaking. Yeah. So modern pharmacy, modern in quotation marks, it's still the same, right? You have pharmacopoeia, you have 1,900 drugs in it, and the doctor looks up for a given indication what drug to prescribe, and then fills a prescription, and you get it in the pharmacy, and you pop a pill. I think that's and Ernst talked about this modern development taking anywhere from 10 to 20 years, and anywhere from $1 billion to $2 billion. In these stages, it's all very well regulated in a pipeline-like process. But what I want to emphasize in addition to all of this is that the success rate in this process is pathetic. It's absolutely incredible. You start with 100,000 potential entities to choose from. Do you know any human activity which is so failure-prone? You know, and you end up with one drug if you're lucky. In fact, 1 out of 10 drugs which end up in the market, they actually don't make any money. So, but there is a miracle on the way. Big pharma makes consistently money, 10% profit every year, no matter how this process performs. And anyway, I think Ernst talked a little bit about this. I don't want to go into the business side of things. I want to go into science. Not quite sure how to. Maybe I can move some of this because.
Sure.
Okay. So where can computers, analytics, high performance calculations speed up the? I'm not saying that we can predict everything by running computers forever, but we can speed up and end up, instead of 100,000 entities to be made, synthesized, and hundreds of millions paid for it, maybe we can end up with 10 good candidates and push them through very quickly through preclinical into phase 1. So in many aspects, I mean, we now have these humongous databases, genomic databases, proteomic, metabolomic, and of course, chemical databases. And we know a lot. In fact, there is software which is incredibly powerful that is based on artificial intelligence. And you can give a chemical formula that you want to test. It will predict for you what toxicity this drug will have in mice, for example, or humans if you want. So, so that's one. Big pharma actually moved towards high throughput screening. So they have robots. We talked about robots today, and robots do this, repetitive work. They can do 1 million compounds to be tested on a target in a week. And I propose that this is actually still a stupid activity that can be done by computers rather than robots, and you don't need to synthesize molecules. So we believe in virtual screening. We believe in partnerships with IBM and companies like that actually are forward-looking rather than backward-looking. and combinatorial chemistry. So instead of synthesizing compounds, you can make them virtually in the computer. We have recently completed a project with the Institute of Virology, the Li Ka-shing Institute in Virology, for a new target that was, first of all, modeled in the computer, and then we created 60 billion chemicals that were screened virtually with IBM's help in 2 weeks. I mean, no pharma in the world, even if they spent all the money they have, you cannot create 60 billion compounds. That's about $1,000 on average per compound for testing. So do the math, 60 trillion. Okay, so this is just some ideas, food for thought. So at all these stages of traditional drug development, we can use shortcuts by computational analysis. Undoubtedly, there is uncertainties because models are models. This is not reality. The real world. And Ernst mentioned the real world solutions. These are not real world solutions. These are virtual world solutions. But I propose we go virtual world to real world, maybe clinical at the very end, and save a lot of pain, save a lot of patients, save a lot of money. So we need to integrate massive amounts of biological and medical data that all of it impacts drug development, especially in the age of personalized medicine. We overuse this word. Actually, there's no such thing, yet. It will happen. I think maybe even in our lifetime, if we live 100 years, 40, maybe even 96 years, 200, whatever. So, DNA, protein sequences, where gaps exist and will exist is in protein structure determination. So crystallography has limitations, and in fact, even if you crystallize the protein, you still cannot rely completely on the structure. You have to do some processing afterwards. Clinical patients eventually have personalized, and I really like this idea of a personalized genome and being born with your DNA fingerprint, and at birth you get all the drugs that you will need forever, already in a virtual form, and if you have money you can afford, you will get them. I would like everybody to be able to do that, but so this creates an explosion of computational data, hundreds of completed genomes. I'll give you one example from the U of A, from our own turf. I worked especially on tubulin, one important protein that is essential for cell division and other things, but from oncology point of view, tubulin has been a target for half a century. And every organism on this planet makes tubulin, or an analog of tubulin. There is no exception. So until Gain Wang came on the scene, and the Beijing genomics project, there were something like 300 sequences of tubulin available worldwide. Gain Wang and his team, for us, created 1,000 sequences of plants, just plants, simply because they had funding for plants. Well, imagine if we wanted to do it for patients. Right? We actually diverted some funds for patients. We sequenced 100 breast cancer patients. Anyway, that was a little bit of a tour de force, but but I think for the benefit of humanity rather than diversion of funds. So the key areas of bioinformatics we have to integrate and bring to bear on the drug development are sequence analysis, and then structural bioinformatics. From sequences, you need physical structures. So that's where physics comes in, and a lot of people ask me, where is physics? Because I'm a physicist, and it's everywhere, but you can't really pin it down to a single thing. This is probably the closest. When you create models of proteins and enzymes in the computer, you use physical laws, actually Newton's laws, and if we really want to know something very, very well, we actually solve quantum mechanical equations for these receptors, for example, and their binding to drug binding to their structures. We can also create shortcuts because in many cases you have sequences, you have no structures. And we can create these models in the computer. We've done it for tubulin and other proteins, actin and so on. Specifically for drug discovery, we have to keep in mind there's 50,000 proteins whose structures are known. About 10,000 human proteins are structurally known. And we have, about 100,000 proteins in our bodies, different ones, and everybody makes them a little bit differently. So the scope of work is enormous, but not impossible. And out of these 100,000 proteins, 1,500 have been identified by NIH as potential targets for drug development. Approximately 400 proteins are identified as targets for oncologic applications, cancer. And only about 80 so far have any known inhibitors. So we are scratching the surface. The amount of work is huge to be done, and I'm so impatient and sometimes upset that we're not throwing huge resources at these problems because all of it is solvable. So there is 103 exactly cancer chemotherapy drugs approved for use in North America, roughly the same in Europe. There's 200 types of cancer. What are we talking about with personalized therapy? I mean, there's a huge disconnect. So this is the enormity of the problem, if you can see. The enormity of the problem is that this is a signaling pathway for cancer, kind of a standard textbook picture. And every arrow that you see is some sort of signaling path. And if you really took it seriously, you'd have to inhibit these pathways. Because this is generic, by the way. Different cancers have slightly different topologies of these pathways, and so if you take it literally, I am actually surprised that we can cure any cancers at all, because we use up to 4 or 5 drugs in a cocktail, maybe 8. I've seen the biggest, but most of this would be against side effects, not really active ingredients for. So it's a systems biology problem. Cancer and maybe other diseases, but I don't know that much about other diseases as much as I do know about cancer. It's a systems biology problem, and therefore we need to develop systems pharmacology to look at it as a problem of changing the dynamics of the system, not looking for a silver bullet. That we will not find one. But we will find inhibitors for every single target. Every single arrow that was shown on that slide can be stopped. Okay, so keep in mind 400 of these arrows in general pertain to cancer. We have found maybe 80, so 20% or so potential inhibitors. We still have 80% to go. And then the question, how do you combine it? You know, the drug interactions, all of that. I don't have the time to discuss it today. I'll race a little bit faster now ahead. So that's the arsenal currently of cancer chemotherapy, about 100, I said 103 drugs. They come into 8 different classes, and we really don't have much to choose from. When you think about the fact that almost every major protein in tumor cells is mutated in some way. So not only do you have to think about the generic target, but also patient-specific phenotype that needs to be dealt with. So for example, one of the key proteins, P53, which is the guardian of the genome, is mutated in 50% of all cancers. All patients have 50% of all patients have mutations in P53, and the remaining 50% of mutations in their partner proteins, MDM2, MDM4. So basically, if you really take it seriously, you'd have to have at least a specific drug for each of these mutations. And for P53, there is hundreds of known mutations. Anyway, the problem grows exponentially. This is what we have put in a cell cycle schematic. So actually, most of the chemotherapy drugs have to do with cell cycle arrest and signaling inhibition. Let me just, in the interest of time, also talk a little bit about the failure in drug development. And almost all drugs fail eventually. If you're a chronic user of some drug, drug resistance comes in, especially in cancer. And failure in drug development are actually very few due to lack of efficacy, only 11% of the failures, but mainly pharmacokinetics, so the drug is either too short a period of time in the body or too long, or some other, or bioavailability, toxicity, especially toxic effects. And so this is another area actually. Toxicity prediction is algorithmic. If you had structures of all functional proteins that are or metabolic enzymes that are related to toxicity, you can do all these predictions by what we call reverse docking. And we do it routinely. We've done sort of simple exercises on doxorubicin and paclitaxel, some of the key drugs, and predicted computationally the known toxicities of these. So again, this is not rocket science. And especially if you compare wet lab screening to virtual screening, computer screening, I call it dry lab screening, the computational advantage is enormous, because even in Novartis or Pfizer, these companies, they can screen 1 million drugs a week or so, and by the robots on hundreds of well plates. We've done 100 billion compounds in 2 weeks. So I'm not impressed by Novartis by any means. Of course, their prediction rate is better because it's real wet stuff, and they tell me, we saw this. I will only tell, so our track record is about 20% probability of accuracy of prediction, 20%, 16 to 20%. So it's not impressive, but it's not because we are so stupid, it's simply because we don't have enough resources to improve the accuracy of calculations. We have very accurate programs, software available, but hardware is limited, and time is limited, and everything is limited. This is our computational infrastructure at the Cross Cancer Institute. We have 2 clusters with about over 1,000 processors, and we use BlueGene at IBM. We got exclusive access to this by a very nice arrangement, where things that take a year or 2, even on these exclusive clusters for us, we can do in 2 weeks in New York, Hawthorne. So the possibilities exist. I'll skip this. This is basically how you go from sequences to structures, and again, a lot of prediction is available. We use, this is where physics comes in. We actually, one word that I want to really emphasize. This is not hokery-pokery. We actually represent every single atom of the protein, and up to thousands of atoms, and their interactions with each other. And we solve up to 100,000 differential equations simultaneously with millions of steps. So the computational firepower that is thrown at these problems is massive. These are not cartoons, although the slides are. So, for example, we worked on tubulin, I mentioned, and we know exactly how each drug that targets tubulin interacts, where it fits, where the bonds are made, how strong this is, how we can remove it, and so on. And then we use something called virtual screening. So if you have a target, then you basically, it's called docking. You dock the molecule to all over the protein. In other words, almost like land a rocket on some planet and find out where the landing is causes the strongest attraction. And this is the site, the preferred binding site. And we can repeat this process millions and billions of times and get better and better statistics and correct answers, and either use existing libraries of compounds. So, for example, if a company like Eli Lilly would come to me and say, we have a library of 500 million compounds, can you screen them for something? And we could do it. Or we create our own libraries, or we access publicly available libraries of chemicals or pharmaceutical agents. If it's, generally speaking, chemicals, you have to be careful about their pharmacological profile, so drug likeness. There are some criteria. Again, I want to emphasize algorithmic predictability. This is very close to being exact science. And so that's basically how the the wheels in this cog turn. And so what I call it contents, what we put in into the software that predicts the existence of novel drugs is the content. So compound data sources, drug data sources, and some molecular toolkits to manipulate this, and computational methods, which are the crudest is called molecular mechanics. The highest level of molecular dynamics. We actually equilibrate the system to find its most energetically favored position. And the most advanced is quantum mechanics, molecular mechanics. So now we can use quantum physics to really represent bond making, bond breaking, with electronic degrees of freedom, not just protonic, not just masses fitting into nice little niches. And then docking software. So I have to admit, most of it is not original. I mean, I would say almost 99% is not ours. But the package, the combination, the pipeline, the virtual pipeline is ours. And so I boldly predict that we will have apps. Maybe we will live long enough. I hope you're right. And I like this idea of having these apps on the cell phone that will give us instant diagnoses and maybe a transmitter somewhere in the body that will release a little pill and case closed, right? Another cancer cell killed. This is I have to apologize if you can read it or not, but there is actually a very well designed process, the flow chart that we follow, like in engineering, in terms of searching for inhibitors from. And on the left, you have basically, you start with databases of chemical compounds. On the right, you have a protein database of targets. What I'm trying to say is that, in principle, the problem can be solved once and for all diseases, for all targets. And I call it the Project Pharma Matrix. And you may have heard about the Human Brain Project, which is now the biggest scientific project in the world. Outside of the existing ones, like CERN, it's in Lausanne, Switzerland, was approved early this year with 2 billion euros from the European Union as a flagship project. And we are official collaborators with Project Blue Brain. They would like to put Pharma Matrix, which is prediction of neurological drugs. They're interested in the brain, using this kind of technology, software. And IBM actually is a strategic partner with Blue Brain, as you know. So this leads to enriching of these compounds and filtering. You have to do a lot of filtering because there's no point in actually running some silly simulations. So we create shortcuts and filtering methods to find out what is garbage and what is worth keeping. It goes on, and that includes optimization and also prediction of pharmacokinetics, toxic effects, off-target interactions, and so on. So, in principle, all of it is doable today. And in 2045, let's say, we hopefully will have a personalized genome for $10, and this will be available to everybody at birth. And I want to add one more thing to it, which is my other project that I've been, if this was not enough, the virtual human. Pharma Matrix is really finding inhibitors for target proteins or enzymes. Virtual human is the creation of a three-dimensional, maybe even personal human, that will allow you to monitor drug distribution, metabolism, and effects in real time. That's also scalable, doable, and the future is friendly, as they say. So thank you very much for your attention.
A few minutes for questions or general questions. I might raise a comment. I've been involved with this with Jack for a couple of years, and this is honestly Nobel Prize winning stuff. It's not just interesting. In my judgment, with 5 decades of experience in this area, the principal problem is not knowledge. It's not it's not technology, it's not capability, it's not competence, it's the inertia of status quo. And if you really look at it, I mean, why is Pfizer not breathing down this guy's neck? Why is Lilly, even though they're going bankrupt, not breathing down this guy's neck? Why is Novartis? You know, why are these pharma giants who, you know, are turning in profits of billions of dollars a year in the most outmoded system in the world for drug development? I mean, it's horrendous. It's obviously decades behind where they need to be in order to do what they need to do with what they're going to have. Why aren't they doing that? And that's a question. I can't answer that question. We've put Jack in front of Pfizer.
Yes.
We've put Jack in, you know, in all, and
Can I give you a partial answer?
yeah, please. Have you got your wallet?
I actually have a bit of, as Jack knows, I have a bit of background in this too. You know, I docked Cerubis into allocation factor alpha more or less by hand in 1989, I think.
There you go.
But as you alluded to, I have a background in 3D structure of macromolecules, protocrystallography. That was my PhD. We did it at the time, as you correctly said, we were too early. Because at that time it was completely and utter nonsense. And it did not, and like many other things in science, that's part of the problem. We overhyped at the time the structure-based drug design, auto-docking, and all these kind of things to the point that people became very gun-shy.
Mm-hmm.
That's part of the problem, in my opinion.
Very interesting. Yeah.
Because
Yeah.
we did it at a time when really the predictive value was somewhat questionable, to say the least.
So
Yeah.
why aren't we creating industry around it here in Alberta?
Why
So
aren't
that if you have some world-class techniques and technology, why don't we do something with it here? Why do we need Pfizer? Build it here.
Okay, I've been trying to say that very sentence.
There's a problem, and the problem is there's a serious disconnect. And the disconnect is that we, through our democratic society, are giving vast amounts of money to governments. And we are starting to be tied to the tit of government in every way, shape, and form. And this company in this province could not make it without the support of government. And we can't get the support of government when they are just talking about a $6 billion deficit and cutting, you know, schools and teachers and universities. I mean, there is a serious structural disconnect between our ability to do things and our capacity to pay. Because we have, and I'm not an Ayn Rand, okay? But the reality is we have given so much of our money and our responsibility to government that they now are what I call the golden man. He who has the gold rules. And we got a real problem.
And can I add something? Just
Yes.
a specific point. And aggravating to that is it is in particular in this area, in this sector of, let's call it big science. You need, at least initially, infrastructure installation that cannot be commercially sustained. That is usually, back in the past, these were supercomputer clusters. But that's not quite true anymore, because these days, the power of some...
Moore's law still
Yeah.
is, you know, like
So-
computing you need, just wait 2 years, right?
Yeah.
You'll have
Yeah.
it, so.
So
Well,
that's-
it's there now.
But,
Yeah.
It's absolutely there now. It's
but
making
that
investments
has
in the
changed,
province
but-
to use it for that
that
and
was-
many other things. I can give you
but it
10
was just
other
a-
examples where
It was just one example.
done
Like back in the
groundbreaking
days
stuff.
when this kind of thing started, we depended on supercomputer clusters and supercomputer installations that were really dependent on public funding. And that's still true for synchrotrons and stuff you need for protein crystal structure determination and things like that. Once you develop a scientific sector, so to speak, around some very large government-funded infrastructure, it is extremely hard to take that sector and take it away and commercialize it into small companies. That has been tried, but it is extremely difficult because you're based out of these billion-dollar investments.
Yeah.
That's an aggravating problem associated with what you just said.
So, I mean, the basic problem we have though on that $6 billion deficit is, I mean, you know, we're so dependent on oil and gas, we need diversification. So what you're doing would be diversification, right?
Look at, I mean, I'm not trying to elevate the European Union, but they have bigger deficits than the EU, and they allocated 2 billion euros to a purely scientific project, the Blue Brain.
I don't think we're going to solve this problem.
No.
Sir,
It's very interesting to hear what you're doing. It's fascinating.
somebody's behind you is asking for a question.
So right now, the biggest road on the block on the road is the lack of structure of proteins to being able to do the docking and
Yeah.
do all the study. Let's pretend that's been solved and we are able to find all molecules or have good targets that are going to be inhibitors for the proteins that are relevant. Now we have to go one step further, and as you very well said, a cell is a system, and this is a systemic problem. And one thing that is very important about these kind of problems is that they have a lot of feedback loops, and these become, then you start to have emergence phenomena
Right.
that you wouldn't be able to predict at all, even with, what's your take on that?
No, I didn't want to go too technical. I thought this was already very technical. But there is actually a solution to that problem as well. We can also simulate the entire cell. And in fact, one of my colleagues at the U of A, Mike Ellison, was again ahead of his time. About 10 years ago, he championed a project called Project CyberCell. And he created a three-dimensional model of the entire E. coli, admittedly, but that was a decade ago. So today, we can create a synthetic cell with all the pathways, interactions, and simulate all of this. Even if you don't want to do it in 3D, you can simulate differential equations that do the substantial amounts of simulations for the key interactions. So the next level, which is the matrix, I call it pharma matrix. So it's drugs and proteins. So it's kind of static, right? Then you add one more axis, which is time, and then allow interaction in protein interactions and drug interaction, drug protein. It just makes it a bit more complex and then definitely more challenging. But we are on the right track, and I don't think there is. Yeah, it is challenging, but I don't think I would give up. Let me put it this way.
Not to discourage anything.
Well, perhaps we should move on. You all have a reception that you're invited to, so let's go. Thank you very much.
Very interesting.